In this section of this series on methylmalonic acidemia, we will review clinical and scientific research delving into MMA. So far, we have learned some background in MMA, its effects, and some treatments that are currently being used. Clinical studies and scientific research are the main sources through which knowledge about methylmalonic acidemia have been gained throughout the years. These clinical studies are designed carefully and evaluate the success of certain treatments or regimens. One way to find a clinical study in which to participate is by going to clinicaltrials.gov. By typing in methylmalonic acidemia into the condition box and selecting the country and even the state, search results pop up about the various trials that have been completed or are ongoing. The status of each trial shows up on the left side. If the researchers running the trial are recruiting, the column will say so in green text, while completed or terminated studies have this status in red text. The National Institutes of Health also has ongoing studies.
The other way that families can stay informed about the trials, of course, is to join the support groups and be in touch with other families, such as those in the Organic Acidemia Association. That's a good way to keep connected to find out what's going on in the research arena. Right now at NIH, we have what's called a natural history study. And that natural history study has been ongoing since 2004, when we were fortunate enough to meet some families with MMA to study what's called natural history of the condition. In other words, what happens to a patient with MMA over time. Overall, it's unknown to the community, for example, how many times a child might have to go to the hospital, or how high their levels might go, or when they might get kidney disease, or if they have other organ involvement. So that's what our study tries to do. We try to understand the connections between the type of MMA a patient might have, their symptoms, and also to study patients that come into our research protocol to see what the effects of treatments might be on them. Not all centers treat all patients with MMA in the same fashion. Right now, there are no interventional trials for MMA at NIH, although we are hopeful in the future that we will be able to offer treatments for patients that are using the new genomic therapies, such as mRNA therapy, conventional AAV gene therapy, and genome editing-based treatments to treat MMA.
We will discuss 3 projects of scientific research that aim at finding a more permanent treatment for MMA. Firstly, there is methylmalonic acidemia mRNA 3704 with Moderna Scientific Research Part 2. This study is particularly aimed at evaluating the effectiveness of mRNA 3704 therapy as a treatment for MMA. For a little bit of background information, mRNA carries a blueprint of proteins that DNA codes for. Inserting the correct blueprint, in this case for the MMUT enzyme in the liver, can help in producing more of that necessary protein.
This is a transient treatment for methylmalonic acidemia M type. It involves giving a tiny little nanoparticle, a lipid encapsulated nanoparticle. Imagine a soccer ball, and on the inside of the ball you have air, but in this particle, the outside of that soccer ball is going to be what they call a lipid particle. And the inside where you'd have the air, that's going to be the messenger RNA that delivers the actual coding part of the enzyme for the methylmalonyl-CoA mutase enzyme. And so the way this works is a patient will get an IV or some other way to get into the venous system, and the patient would get an infusion. And this infusion contains some fluid, probably some saline type fluid, and it contains these particles. And the particles flow throughout the blood, and they land mainly in the liver. And when they get to the liver, they unload the inside of that soccer ball, which is the mRNA part of it, into the liver cell, the so-called hepatocyte. And then what happens is that mRNA encodes for the enzyme. And then the enzyme is made, and it is transported into the mitochondria where it normally needs to be to work. And then all of a sudden now that cell is basically corrected. It doesn't have MMA anymore. It has normal mutase enzyme in it.
Though this therapy is transient, this could be an advantage, as patients could stop taking the medication if harmful side effects appear. It could also be accessible to some patients if large batches could be made at one time.
The mRNA therapy does produce the enzyme in animal models of MMA. It shows that it will direct the enzyme to be made in the liver. And that is a place where we know this enzyme must be made for therapeutic benefit in mice and in humans. If a patient was going to get this infusion, they're going to have to get it on some sort of a schedule, in theory, their whole lives.
Secondly is a study by Logic Biotherapeutics called LB001 for the treatment of methylmalonic acidemia. Unlike the mRNA 3704 therapy, this study involves direct edits to the genome of the recipient. The LB001 is an adeno-associated viral vector, or an AAV vector, that has the correct version of the MMUT gene. The vector will incorporate the functioning MMUT gene into the genome of the patient.
These AAVs are benign viruses in humans that were reconfigured for recombinant DNA use by removing all the viral genes except only the very ends of the virus, the so-called inverted terminal repeats or terminal repeats, that the virus needs to package into the viral shell. And so with this method, what one does is the virus is configured to have DNA sequences that flank the end of the albumin gene. The albumin gene is found in all cells, but it is highly expressed in the liver. And so this AAV, the way it's been designed is it has an ability using the natural recombination machinery of the cell, so-called homologous recombination, to insert a copy of the methylmalonyl-CoA mutase cDNA precisely at the end of the albumin gene, right before the so-called stop codon of albumin. So what happens is, under the conditions of a successful integration event, and this, what one must say here is that the probability that this will happen in every cell, this type of event I just mentioned, is very, very low, because natural homologous recombination is a low frequency occurrence. But then the cell will make the methylmalonyl-CoA mutase enzyme under the control, so to speak, of the albumin promoter. So it will make very high levels of the enzyme in the cells that are corrected. Now, studies in mice that have been going on in my laboratory suggest that a cell that is corrected has what's called a growth advantage. So it grows a little bit faster in the environment and probably lasts longer than a cell that doesn't have the correction.
The growth advantage may allow these corrected cells to grow better than uncorrected cells and make for increasing numbers of the enzyme over time. The project is in its earlier stages, having just ended the second quarter on June 30th. It will likely extend further than the end of 2020, if not longer. The last kind of treatment involves canonical AAVs. These have proven to be very effective in mice and look promising as a way to treat patients.
The last type of a product that, again, is a gene-based product, it would be a canonical or a conventional AAV, which I believe there are companies that are interested in working on this approach as well. This latter approach that I just mentioned, so-called canonical AAV or conventional AAV, is very, very effective in the mice. It's probably the most effective preclinical therapy that we have in the animal models.
These projects and more make a cure seem possible in the near future for MMA.
Right now we're on the verge of seeing a number of new treatments get to the clinic. I think we will see patients start being treated in the next few years, and it'll be of great interest to everybody, especially the patients and the families, to see if these treatments actually really can improve the patients, their quality of lives, and their metabolic phenotypes.